Queen Relish has told me many times that she loves this topic, and it is on these. Not at all what any of y'all were thinking. The tiny crab, actually. The tiny crab. Look at that pretty, beautiful, cutie patootie little crab. Putative nociceptive responses in a decapod crustacean. The shore crab. Why didn't they just say pain? Hannah Counter, that's a great question. That is because pain and nociception do have differences.
What's the difference?
And let's cut into the differences between pain and nociception to dissect this out. So nociception here that these crabs are having. So it's just demonstrating that this initial stimulus here can be detected. But and the same thing with fruit fly larvae, right? Insects have that ability to detect it. But is that actually pain? Or is just the very beginning of the detection of that something happened to me? And so that's where it is. It's like I can respond to this external cue. That doesn't mean that they have necessarily pain. It just means that I'm able to perceive that something has interacted with me that could be a damaging thing. So like with the fruit fly larvae, they get touched with a heat stimulus and they go, ow! Yeah, they zip away from the heat response. But that doesn't mean that they actually are feeling an element of pain with that. It just means that they are detecting that there is something here that could be affecting me on a stimulus kind of level. So it's just a part of the pathway versus the entirety of the pathway is pain. So you need nociception to have pain, right? If you have no nociception, you will have no pain. But if you only have nociception, it's just going to be an instant response to the environment, and there is no interpretation of that signal as pain. So that's the difference where in flies, researchers have identified nociception neurons, right? And nociception-based stimuli, like neuropeptides that are released that are the same as in people. So you can study the beginning point of it. And then for Saphira's mama, like how she's asking about chronic pain, the next step is figuring out how that nociception, right, is transmitted to these other elements here and how you can block the other elements. Because you don't necessarily want to stop the feeling, right? You don't want to block nociception, because if you block nociception, you're going to touch a hot plate and have no response to it, right? And then you'd be damaging your body. So instead, you need to figure out a way where you can have nociception, but block the sensory, like long-term sensory put to it, right? So it's that bit of extra information that you have to identify. We don't know. They have the nociceptive neurons, like we know that they have those neurons that are innervating at the site of a negative stimulus, and then that is transmitted to the brain, but it's those next steps that we don't know yet. And even though this video makes it very clear cut that yes, crabs feel pain, so Sparky, the paper looks at the beginning set of neurons. So this video is much more of just this initial like observation, and then the paper is actually getting into a little bit more of the like neuroscience behind it. The neuroscience behind it here only looks at the nociceptive neurons, not the entire picture quite yet. You need the entire all set of neurons before you can really get at if it's really pain, how you're processing pain with like the negative reinforcement, the memory of it, the flight or fight response being activated, right? There's a lot of those additional components that need to be looked at to really get at if it's pain or not. It hasn't been done in the crab, nor has it been done in the fly. So Sparky, I would argue here, this is not yet demonstrating that there's pain, right? The argument that she's presenting is there's brains that are complex. That's fine. That doesn't necessarily mean any of that. The brains are decentralized, meaning that there's small subregions closer in the limbs, kind of like an octopus. So Neil, it's not the small brains that for a quicker response, it's the physiological differences. Like the fact that there is some like immediate brain tissue in the legs mean that there could be a faster response. But this study didn't delineate between what type of neurons are actually detecting it. So if it's just that the nociceptive neurons are getting turned on and that's it, then they're, yes, detecting that they've had a stimulus that could cause damage. But by our definition of pain, that's not pain. That's just they're responding to an environmental pressure on their body, which I know it sounds like, well, how is it that it's not pain? Is that not what pain actually ends up being? It is an extremely complicated response where it's kind of like the definition of a species, Sparky, where it's this vague idea of these are the criteria that you need to have in order to be said to be a species. and then that's pretty much it. Versus in this scenario, with they're not even having all the criteria. They're just be like, oh, there's a negative response, and we don't know. Yeah, exactly. Is it Neil just a negative response, and I'm moving away from a stimulus, right? That's what the nociception is. If I poke your fingertip with a needle, or Andrea No Art rubs a balloon on your butt and you pull away, is that a perception of pain, or is just something like, oh, this doesn't feel good, let me just move on? Smaller brains work the legs. I don't think we know well enough yet, Sparky. The neuroscience in crustaceans is very behind, primarily behind because there's not a lot of genetic tools available for these creatures. There is just a very small group of researchers who have them. They primarily are studied for lifespan differences and not so much the neuroscience side of things. Although we'll take a look at the paper where they're actually starting to look at some of the neuroscience elements. But yeah, it's an interesting one. It's an interesting one of that pain experience again. The paper that we're going to be looking at today, which here, let me go ahead and bring back up. This one sets to define if the animal can feel pain based on a very methodical level of molecules. And one is the presence of nociceptors. Those are the receptors on either the exoskeleton, if it's like the crabs or the insect, on your skin, that are that initial response point. If you don't have those, then they're just not going to be feeling in the same kind of what we're used to studying. And so they looked at the central nervous system and its connection to these nociceptors in these crabs. And they looked at two kinds of stimuli. One is mechanical, so by poking the crab, and the other is a chemical by acetic acid. And they actually found 32 different areas on the crab that had a response. They actually were able to map out where on the crab there's actually feeling with this, which I think, again, is really, really cool. But there's been previous to this paper, there's been no looking at electrophysical studies. So, like, no actual link to neurons, just that if you poke them, they might move. But here it's like a legitimate way of identifying if there is pain and this kind of sensational ability. So they saw in the soft tissue that there was some kind of pain receptor. So, like, Hanaconda identifying if they have these certain kinds of receptors, how you should change your own behavior when working with them. I feel like that makes it very difficult because, like, when is the right and a wrong time to be able to hit that? Yeah, I feel like it's a tough one on that front, but it's still really, really interesting. They found, again, in the soft tissue that there were neurons that fired when, like, using either this mechanical stimulus or the chemical one. They see a spike in brain activity, which I think is, again, really interesting that they suggest it's probably for danger avoidance that this evolved. Yeah, I thought it was interesting because it's a new model in which there is this sensation of pain. Again, pain's been studied in insects for these nociceptors, and that's actually why researchers were able to study this in the crabs. It's all based on existing data that they've collected from the fly models. Because they're just so similar in the fly, it's near identical of what you're identifying in the crab as well. We can quickly look at the figures too here. Yeah, y'all, it was kind of neat that they had this ethical consideration put in, which was, again, really interesting. I haven't seen this before, especially. They even point out, like, the European directive doesn't protect these animals. They're like, well, we went above and beyond trying to make sure they were not experiencing pain, or at least, to the same level as you could have. And then here is the experimental setup. This is where these dots are where they did mechanical stimuli and where they did noxious stimuli in the form of chemical as well. So where they were trying to identify where the stimulus could come in and then be processed by the brain. And so, again, the red dots are chemical hits, and the black dots are mechanical, and they're using a certain kind of hair to do that, basically tickling the animal. Here, Sparky, you asked about what the brain looks like. Didn't get too much bigger. That's the brain here. So it's got the main body of the brain up here, and then it spreads deeper down, and then it has that more decentralized. So again, this is kind of reminiscent of the nociceptive stimuli, where it's more in, like, the legs or, like, other peripheral nervous system elements that allow it to react quickly to that nociceptive stimulus when all of a sudden it comes in and touches its arm, whether it be through chemical or mechanical sensation. And here was just the way they did the experimental protocol. So either they applied, the chemical cue or the tactile cue They then had a 10-minute wait time, and then they put them into the box where they were video recording the application of the other kind of cue. So here it's the chemical cue added, then they added them into the water for a little bit to acclimate. They then take them out, and then they do the little hair on the body to see where they respond with a high-speed camera as well. So again, like a neat, clean-cut way of doing the experiment. I guess as far as you can do it with these crabs, there's no really genetic trick that they can be using for something like this, at least not yet. So that was the outline of the test. Here are the electrophysiological responses. So remember, they actually hooked up an electric rod to measure the neuronal firing. So again, nociceptors, the chemical stimulus that they used was acetic acid. Placing acetic acid on the shell of the animal caused there to be a reaction. They then also used mechanical signal, which was a really thin hair, and that's what triggered like the touch sensation on these animals. They got the recorded activities. And again, here is the signal strength that they got from these neurons that they were recording as well. Like there's the voltage measurement and the different treatments as well. So they saw, as you would normally see with the increase of the chemical cue, the higher the neuronal firing was, and a similar one when you're looking at the mechanical stimuli as well. Basic analysis, but it's the first time, and it's also pointed out in their discussion. To our knowledge, this study is the first to report the in vivo electrophysiological recordings of the nociceptive responses in crabs. Detection of pain, again, with different definitions. In this paper, they were looking at the very, very molecular and neurological level, which I think is really cool to be able to dissect. And then the video is more of that broad definition and kind of going in between which one is appropriate. And it depends, honestly, on what kind of question you're asking. And that's the thing, too, like, boy, that they're saying in the paper is, like, you can tell that the neurons necessary to detect that stimulus are there, and they are detecting the stimulus, but it doesn't necessarily mean that they feel pain, right? It's just that they're detecting the stimulus. So the next step is then, okay, figuring out what those neurons connect to in the brain and where else they're taking the information to. If there is this extra downstream information that's being transmitted, that's where the pain centers are going to lie. In the crabs and other animals as well when you're studying it. But again, it depends on how you're defining pain. Exactly, Void. 100%. That is true. That is very true. Is damage avoidance or reaction dominance on the molecular level? Damage avoidance is nociception, and that is different than pain. So if I poke your hand with a needle and you pull away actually before you recognize that was a pain stimulus. If I can actually shut down receptors in your brain that will tell you that that's pain, and you'd still be able to pull away very rapidly. So then on the processing level, the damage avoidance comes first, and then comes the identification that it's pain. Does that make sense? That's nociception right there. So dominance, that's the first thing that happens in terms of what molecules are activated and the speed of it. So you get that reaction first. You're not reacting to pain, you're just reacting to there being this potential damage stimulus. And the motor neurons are what fire on. If that was it, we wouldn't feel pain. The pain processing takes longer because it has to go to the brain and get processed. It's also processing in a couple of different regions in the brain. So, Sparky, the difficulty is if you're defining pain on a molecular level, where it's like these neurons have to be present and these molecules have to be present, you have to look at the animal first, right? You can't assume. And so that was, I guess, the difference here in the crabs is that behaviorally, they had identified a lot of reasons why maybe these animals felt pain, but no one had looked at it on the electrochemical signals, like within the neurons themselves. And so it just depends on how you are going to define that pain and what is your threshold of definition. And it's kind of wild on like how sometimes vague these definitions can be. Like I always been like, oh, it would totally make sense that this is a very easy thing to define, but it seems like it's very, very not easy to define and it's extremely vague. Doesn't this complicate everything, Sparky? But right, this is a further complicating factor. If these are all some things that are like the precursors to having actual pain sensations, that's not proven here, right? They didn't prove that there's pain sensation. They just prove that there is this nociceptive response. right? Is that sufficient to yet interpret pain? Probably not yet on the molecular level, but if you tie it in with the behavioral one, it's like, uh-oh, now there are all these ethical concerns coming in with working with these animals. So again, this is the first example in terms of the paper of there being this detection of nociception. Again, they looked at, just a little pullback, they looked at 32 different areas around the crab's body that identified, yes, there was these nociceptive responses. They looked both chemical as well as physical sensation, demonstrating that yes, these animals have this nociceptive neurons. They did the electrophysiology in the animals to show that there's peaks from those neurons, and it is in the soft tissue. They don't know where the neurons go in the central body of the brain. They don't know if then there's this memory formation the way it's identified in other animals, but there is the foundation of pain sensation in these animals established in this paper. And that's a really, really neat study with a lot of ethical implications as well.